Welcome to EAG Laboratories webinar on thin film analysis. I'm Monica Newberger, Manager of the Surface Science Group at EAG's lab in Sunnyvale, California. Over the last 15 years, my career has been focused on surface chemical analysis and materials characterization. My particular areas of expertise are X-ray photoelectron spectroscopy and time-of-flight SIMS. For the purposes of this presentation, we will define a thin film as a layer of atoms or molecules ranging anywhere from tens of angstroms to hundreds of nanometers thick. As a point of reference, gold foil can be roughly a half micron thick, which is equivalent to 500 nanometers or 5,000 angstroms. These films can be decorative, but they are often functional, providing protection against contamination and wear and tear. They can also be used as electrodes, transport layers, and in many other applications. Understanding the properties of these films can be critical to understanding and improving the performance of your product. There is a huge variety of techniques available for materials characterization. Here at EAG, we've organized them into what we call the bubble chart. The vertical axis shows the detection range of each technique in atoms per cubic centimeters. The horizontal axis gives the analytical spot size or analyzed area of the technique. Inside the box, we have surface analysis techniques, which have both dimension and detection range. Red bubbles indicate techniques that provide chemical bonding or molecular information. Above the box are imaging techniques in green. They don't provide compositional information, so the idea of detection range doesn't apply to them. However, their lateral resolution or spot size is very important. Energy dispersive X-rays, or EDS, can provide atomic compositions, which is colored blue to distinguish it from techniques providing molecular information. On the right-hand side of the box are bulk techniques, which provide concentration with no spatial resolution. We will not be discussing any of those in this webinar. Of the many techniques available, the ones colored here are those that will be discussed in this presentation. I'll discuss these techniques in terms of sample applications. We'll start by combining imaging techniques with compositional and structural analysis in an R&D project for LEDs. We will then look at two types of chemical analysis, one for an inorganic film and another for an organic layer. The first LEDs used semiconductors as light emitters, but perovskite structures can be cheaper and easier to manufacture. They are also strong light emitters, which can be made into a wide variety of colors. The photo on the left shows the test device with its schematic diagram on the right. In between the glass substrate and the top metal electrode, we have a transparent electrode, an electron transport layer, the perovskite active layer. The whole transport layer was intentionally left out for this study. The questions we will address are the composition and uniformity of the layers. For that, we will use TEM EDS. We will use XRD to determine if the active layer had the correct perovskite structure. And we will use SEM and conductive AFM to investigate why some devices were not lighting up. Here is a schematic representation of scanning electron microscopy, or SEM. An electron beam is scanned over a surface, exciting a certain volume of the sample. Detailed images are generated by collecting the secondary and backscattered electrons. The X-rays generated from the sample are indicative of the elements within that volume. The lateral resolution of SEM images is on the order of nanometers, and the X-rays, or EDS, can come from a micron deep. You can also see that the lateral resolution of the X-rays are greater than that of the electrons. Some films are too thin to have their thicknesses measured in an SEM cross-section. Also, a thin film stack can give the same EDS spectrum as a homogeneous mixture. Transmission electron microscopy, or TEM, provides finer resolution than SEM. TEM analyzes electrons transmitted through a thin prepared cross-section. Its lateral resolution is on the order of angstroms, and the EDS resolution is also finer since there is no longer any bulk to the sample. So here we have a TEM image of the cross-section of the LED test structure. The main contrast mechanism in this image is atomic number. The bright band near the top is the metal electrode, followed by the active layer. Remember, this sample did not have the hole transport layer. And that active layer is followed by the electron transport layer, the transparent electrode, and the glass substrate at the bottom. The dark areas in the active layer show us there are voids within the film. The EDS map shows us what the layers are composed of. Indium, tin, and oxygen in the transparent electrode indicate ITO. The top electrode is gold. Lead and bromine are in the active layer, and we know that the intent was to create formidimium lead bromide with the formula shown. The EDS map alone does not confirm that the active layer has the perovskite crystal structure needed to create an LED. For that, we used XRD. X-ray diffraction can identify crystallographic phases of a material both qualitatively and semi-quantitatively. It can also determine crystallite size and percent crystallinity. In the drawing on the right, we see the X-ray beam coming in from the left-hand side of the sample. Crystalline materials will produce distinct diffraction patterns, and the sample and/or the detector are scanned over multiple angles to collect all of these peaks. The resulting spectrum is matched to a reference library. This is the XRD spectrum from the active layer, showing that it has the correct formidimium lead bromide perovskite structure. The small lines at the bottom of the peaks show the locations of peaks within the reference spectra. I mentioned that some of the test structures were not lighting up. So the surface of the perovskite layer was analyzed with SEM to try to understand why. These bright features were found, which might be secondary crystal structures. In the higher magnification image on the right, you can see that these features appear to be thin. Some protrude from the surface, and they sometimes form at grain boundaries. This was an unexpected finding, and we employed a lesser-known variant of atomic force microscopy to investigate the possible relationship between these features and the electrical properties of the film. AFM is most often used to measure surface topography, but it can measure many other film properties as well. In the schematic diagram on the right, we see a tip being scanned across a surface. The tip is not always in contact with the surface. There are many modes of operation. Changes in the tip height are measured with a laser photodiode pair at the top of the diagram, and a feedback loop maintains constant cantilever deflection. For electrical measurements, as in this example, a metal-coated tip is used, and it is plugged into a current amplifier. The image at the lower left shows the topography of the perovskite surface, and its resemblance to the SEM images is clear. The brighter colors in this image correspond to higher features. The image in the upper left shows a conductivity map of the same area of the surface. In this image, brighter colors indicate higher conductivity. The image on the right shows a line scan of both the topography and conductivity images taken along the vertical blue line. As you can see, the tall features are not conductive. R&D work on these LEDs is ongoing regarding the composition and structure of these features, but we are going to move on to our second example. Solar cells. Like LEDs, solar cells employ multiple layers of thin films. We will be looking at a transparent conductive oxide being developed for use as an electrode for solar cells. Several different materials can be used as transparent conductive oxides. The previous example had an ITO electrode, and in this example we will compare the relative thickness and composition as a function of depth of two films of aluminum-doped zinc oxide. We will investigate these properties with X-ray photoelectron spectroscopy in a depth profile mode. XPS is a chemical survey technique which can also provide bonding information for the detected elements. It is quite surface sensitive with an information depth of approximately 5 to 10 nanometers. And by alternating data acquisition cycles with ion sputtering, depth profiles can be generated. In XPS, a focused X-ray beam hits the sample surface, causing electrons to be ejected. Only electrons from the top 10 nanometers or less escape the surface and reach the detector. The energies of these electrons tell us what element they came from. They also provide information as to what that element was bonded to. Depth profiles are acquired by alternating data acquisition cycles with ion sputtering cycles. Monatomic ion beams such as argon are ideal for profiling inorganic films such as these thin conductive oxides. Sputter rates can be varied with beam energy. Bonding information is sometimes available within a depth profile, but it is important to know that ion bombardment can alter both the composition and chemistry of a sample. In a later example, I'll show how gas cluster ion beams can be used to profile organic materials. These clusters can contain tens to thousands of atoms which all hit the surface together. At impact, the cluster breaks up and its energy is dissipated among the constituent atoms. Since each atom hits with such low energy, there is little to no surface damage, and organic molecules can be desorbed intact. In the left-hand image, we see the XPS depth profile of an aluminum-doped zinc oxide film on silicon, and we can see that its composition is constant with depth. The depth scale is not accurate since the beam's sputter rate was calibrated on SiO2, and different materials sputter at different rates. As is, this depth scale is good for making relative thickness comparisons. If an accurate depth scale is needed, the film thickness could be determined with a cross-sectional measurement by SEM or TEM, perhaps also X-ray reflection. We see a spike in the oxygen trace right at the interface between the thin film and the silicon substrate. In the plot on the right, we see the individual X-ray spectra for each layer of the depth profile. We can see that the oxygen spike is due to the native oxide at the silicon surface. Silicon, as SiO2, appears at a higher binding energy, which is further to the left than the main peak for silicon as elemental silicon. Now this film was roughly 100 nanometers thick. The data interpretation became a little bit more complicated when we analyzed a much thinner film. This film was roughly 30 angstroms thick on an SiO2 equivalent scale. It also has a much lower level of aluminum than the previous example. The peak for silicon as SiO2 was clearly distinguished from the elemental silicon peak over many sputter cycles, so it was plotted as its own component in the depth profile. At first glance, it might appear that the AZO film has become incorporated with the native silicon oxide. However, this is not necessarily the case. The information depth of XPS is approximately 50 to 100 angstroms, much thicker than this film. XPS can't help but detect the substrate even in the first few cycles of the depth profile. A technique with greater depth resolution, such as dynamic SIMS, or a cross-sectional measurement with TEM, would be needed to investigate the degree to which these layers might be mixed. The examples we've discussed so far have been elemental and inorganic films, but organic films are also a part of our everyday lives. Many touchscreens have an oleophobic or oil-repelling coating on their surfaces. These coatings tend to be fluoropolymers. In this example, we will investigate a visible blemish found on a smartphone screen. This appeared to be a surface defect, so a technique that could identify organic molecules with trace detection limits was needed. That technique is TOF-SIMS, time-of-flight secondary ion mass spectrometry. TOF probes only the top 10 to 30 angstroms of a sample surface, and it has extremely high mass resolution, which allows for accurate identification of even high-mass molecular fragments. It is capable of producing chemical maps of surfaces, and like XPS, it can be used in a depth profile mode. To explain how it works, the diagram on the right depicts a sample with a combination of elemental and molecular species on its surface. A focused, pulsed, primary ion beam, shown in black, hits the sample, causing species in that impact area to desorb. The desorbed species are brought to the same energy, and the time it takes for them to fly to the detector is measured. That time can then be converted to a mass scale, allowing the species to be identified using the equation energy equals 1/2 mass times velocity squared. Here on the right, we see a section of the mass spectra from the blemish area in red on the bottom, and a good or control area of the screen on the top in blue. In the good area, we see several fluorocarbon molecular fragments. These are from the oleophobic coating. We see some of those same fragments from the blemish area, but there are many additional peaks noted with red triangles, and those are attributable to amines. The mass resolution of TOF-SIMS is so high that those peaks can be positively identified as each having the formula CXHYN, as opposed to being a pure hydrocarbon or perhaps even an oxygen-containing organic molecule. We wanted to know if these amines represented an organic contaminant that was covering the oleophobic layer, or perhaps replacing that oleophobic layer. This required the gas cluster ion beam profile I described a few slides ago. These profiles show ion intensity on the horizontal axis on a log scale. TOF-SIMS data is not quantitative, but relative comparisons can be made for the same molecular fragment on similar substrates. The profile from the good area on the left shows a high surface concentration of fluorocarbon, the C2F4 fragment, and that is slowly removed with increased sputter time. We see something similar from the blemish on the right in the way that the amine fragment C3H8N is removed with increased sputter time. What we don't see from the blemish profile is a rise in the fluorocarbon peak after the amine signal has been reduced. This shows that the amines on the surface are not a layer on top of the oleophobic coating. Rather, it shows that the oleophobic coating has been removed and the amines have replaced it. In this webinar, I hope that I have demonstrated the importance of materials characterization in the development and optimization of thin films. Imaging techniques can produce clear and easy to understand pictures showing thickness, uniformity, and many other properties. The examples in this presentation included SEM, TEM, and AFM. Chemical analysis techniques give compositional information over a wide variety of analysis depths and detection limits. The type and degree of structural information also varies with the technique. The examples in this presentation included EDS, XRD, XPS, and time-of-flight SIMS. Other techniques which can also be useful for thin film analysis include FTIR, Raman spectroscopy, Rutherford backscattering, and Auger spectroscopy. The right combination of techniques can give the greatest understanding of any situation you may be presented with. I would like to thank Renee Sully and David Fryoff of the University of California at Santa Cruz for their contribution of materials and providing technical assistance for this presentation. Contributors from within EAG included Arwa Janwala, Yulia Grossman, Wes Naveen, Sarah Ostrowski, and Jinyi Zhang.